Why we are interested in recombinant DNA technology. The reason we chose the recombinant DNA technology is because it's a form of genetic engineering that plays a very big role in the world of medicine and science today. This technology has proven to be important in the production of vaccines and protein therapy such as interferon and human growth hormone. For example, human insulin is one of the first therapeutic proteins that was genetically cloned. The drug is now used to treat diabetes. Recombinant DNA technology allows us to learn more about the human genome, opening up numerous opportunities to create possible treatments for currently untreatable diseases and health issues. We are interested in this topic as future nurses because it's technology that could possibly find a solution to saving millions of lives.
These new recombinant DNA techniques have been found to be faster and more predictable than ever before. Recombinant DNA technology has been used for many years to insert multiple copies of genes into other organisms to give them new traits or to aid in others, such as diabetes. For many years, humans used insulin from cows and pigs, but with this technology, scientists can now synthesize human insulin. In combination with the Human Genome Project, this technology can also be used to detect genes that carry heritable diseases such as cancer and Huntington's disease.
Before this new technology can even be used, we need to decide on an appropriate organism to make it with. So the insulin, for example, used to be made from pigs and cows, but now we're able to use humans, which makes it safer for humans. And so once we have this organism picked out, we need to actually make the recombinant DNA, which is shown in the diagram below and which we talked about earlier in the presentation. So just as a summary, we take DNA from two different sources and combine them by splicing in a section of DNA from one organism to another. And so we can see that on the diagram. On the right, we have our gene of interest, which is called the donor DNA. And we isolate it and then cut it out with restriction enzymes. And then we combine that with genes for antibiotic resistance, which is shown on the left, to make modified plasma. Then once we have this, we use a fermentation tank so that it can replicate. Then we purify the protein to concentrate it and make sure that there's nothing dangerous that we're going to be putting into the patient, because obviously patient safety is first. And then once we have the substance, we're able to put it in the medicine bottles and sell it. So even though this type of technology has been around for a long time, this specific machine is new and expensive, which is why, as in the diagram on the right, it's mostly only used in developed places. So as you can see, about half of the cases are used in North America, then a quarter in Europe, and then Latin America and MEA you can see are very small. So clearly it's expensive and it's up and coming, but it's going to have a great future.
Recombinant DNA is important to nursing because in order to take care of a patient, we need to not only understand their condition, but also understand the patient's genetic and genomic information. This approach should allow providers to prescribe more specific and individualized treatment and to avoid adverse drug and treatment reactions. A basic knowledge of genetic terms and concepts and an understanding of genomics can provide nurses with a foundation that will enable them to provide competent, personalized healthcare.
This DNA technology is used outside the healthcare world as well. Recombinant DNA technology is used to produce foods in the form of GMOs, which are plants or animals that have been genetically modified. This is done by putting a small amount of genetic material from one organism to another. Crops are often genetically modified. Some examples of them are potatoes, corn, and sweet corn. The plants are modified so they're protected from pests and tolerant to pesticides. The FDA does regulate GMOs, but considers the modified corn to be just as nutritionally beneficial as normal corn.